Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we'll be talking about microbial source tracking. This problem came about when I had a bunch of samples from a collaborator, and they were interested in knowing where samples from hospital environments, various wet lab surfaces like the bench or parts of the scissors and things like that, where those communities had come from. And we knew that we could characterize communities in some of the potential source environments, such as gut and soil environments, oral environments, skin environments. And then we had all of these indoor samples, and we wanted to know what were the contributions of the source environments to the indoor samples. So you can do an unsupervised ordination plot like this and see that, okay, they kind of tend to be similar to the skin samples and the oral samples, but some of them look a little bit more like gut. But it's a very qualitative approach. And we wanted to answer this question: where did the indoor surface communities come from in a quantitative way? So we looked at this as a community-wide microbial source tracking problem. And if you Google microbial source tracking or look on Google Scholar, you'll see that 99 out of 100 articles are about individual bugs as markers for fecal contamination in water. Whereas here, we want to look at the whole mixture of one community and another community and try to figure out what percent of the sink community is coming from each of those sources. And we could potentially have multiple communities that are mixing together. So, and what we're interested in are these mixing proportions. So it's a much harder problem because it's a mixture of mixtures. And here's a little demonstration of what this looks like. So we could have 3 potential source environments. This could say be skin contamination, soil contamination, fecal contamination. Each of those is a mixture of different species. And some of the species may actually be the same across the environments, more or less, just in different ratios. And then what happens is there's some mixing event, and we end up with a sink, what we call a sink sample, that has received the mixture. And so now it has a mixture of those same species, and our goal is to recover the proportions of the sources, remembering that each of these proportions represents a different whole mixture of species. This would be very easy in the case where we knew all of the mixing environments, because we could just treat it as a regression problem where we know the fraction of each species in the different source environments. So that's the ST matrix. Then we know the resulting mixture of species in the sink environment. That's Y or Y hat. And then beta is just the mixing proportions. So it's a simple linear regression problem. We try to minimize the sum of squared errors between the S times beta, and which gives us our fitted sink sample and the actual sink sample that we observed. So this is one naive approach. The other work, the most sophisticated other work that had been done was using naive Bayes to classify which community came from which, but this isn't really a mixture model. So naive Bayes is trying to predict the likelihood that the entire community came from one community or the other, not figuring out what fraction of the community came from those 2 different sources. So we ran some simulations, and using these 2 previous or simpler approaches, what we found was that when we made a bunch of fake communities that were almost the same or basically identical. And then what this means is that they have no Jensen-Shannon divergence. That's a measure of the difference between 2 probability distributions. So on the one end, we have 2 communities that are identical, and then we varied how much they differed from one another all the way to the other end of the spectrum where the two communities had no species in common. So the species in community 1 were completely different from the ones in community 2. And at this point, the two source communities have a Jensen-Shannon divergence of 1. And then we were able to tune this knob back and forth. And at each point, what we did is we simulated these communities with varying amounts of overlap. Then we mixed them together at different proportions and ran them through a least squared regression or a naive Bayes classifier to see how well that method did at recovering the true mixing proportions. So what do we see here? On the far left end of the graph, where the communities are identical, both of the methods do really poorly. They have high error. So here we're just showing the error of the method. Then as you start to make the two communities slightly different from each other, the linear method actually does really well right away. So even when the communities are just a little bit divergent, it starts to converge on the right answer of the mixing proportions pretty quickly. Whereas the naive Bayes has very poor estimates of the mixing proportions all the way down until you get to the point where the communities are literally completely different. So this is fine in the case where you know all of the source environments, but in practical cases, there are often going to be other environments contributing. And so you may not have sampled all of the potential source environments. You may not know what they all are. And in this case, the problem gets much harder. So here we have a mixture of mixtures with an unknown source. And I'm showing you the same example of source 1 and 2, and then we have an unknown source here. Then we have the sink sample that's a mixture of those same species. And here the goal is to recover the correct mixing proportions of source 1, source 2, and unknown, and at the same time to recover what is the mixture of species at in the unknown environment. And that's the tricky part. So it turns out that if you run these two naive models on the environments where everything is known, or the mixtures where the source environments are known, they do well, as I showed before. But when you introduce an unknown environment, so here it's the same sources, but there's 25% of some randomly generated unknown environment, they both bottom out at a pretty high error rate. So they don't do as well because they basically can't account for that unknown. So what I realized was that we could borrow an approach from machine learning, which is called latent Dirichlet allocation. It's actually, so it was originally used for topic modeling, which is where you have a whole bunch of documents, say like blog entries on the web, and you want to naturally discover which words go with one another in conversation topics. So I realized we could do the same thing here and use a Markov chain Monte Carlo approach to explore the distribution of assignments of species to source environments within a given sink sample. And what happens here is that the way that we do this is we basically start with all of the sequences that we see in the sink environment. So each of these is an actual OTU, let's say, one observation of one OTU. We pick that OTU out, pretend that the rest of them are assigned to the correct source environments, even though we start with random source environments. And assuming that the rest of them are in the correct source environment, it's actually very easy then to calculate the probability that this one came from unknown or source 1 or source 2. And so we just roll a weighted die and put it in one of those depending on how likely it is to end up in each one of them. So then we put this sample back in, and we pull out another OTU. So then we pull out another OTU and again, pretend the rest of them are correct, but we started off with random assignments, so they're still pretty wrong. And given that the others are correct, it's very easy to figure out the probability that this one came from the source, or source 1 or source 2 or unknown source. So we drop it into one of those bins, stick it back in, take another one out, and we keep repeating this process over and over. And it turns out you can prove statistically that these will converge on the actual distribution of true assignments from the different source environments. So it's quite simple to implement. You literally just assign each taxon to a different random environment at the start, and not each taxon, but each observation of each taxon in the sink sample. And then you can calculate the probability of that observation of that species, or OTU, coming from a given source, given all of the other assignments, which are random at first. It's just this equation, which is the probability of the current probability of that taxon in that source environment, and the current probability of that source environment in the sink sample. And then you just keep iterating on this and updating it every time. So anyway, we simulated the same data and ran it through Sourcetracker, which uses the latent Dirichlet allocation. And what we find is that when there are no unknown environments, it does just as well as least squared regression, which means that we just spent a whole lot of computational effort for no benefit, because least squares works just fine. But when we go to the unknown environment, where the other two methods fail, Sourcetracker actually can recover the mixing proportions of species from the unknown environment, and therefore also the mixing proportion of the unknown environment in the new sample. We applied this to several different types of samples. Some were from hospital surfaces in neonatal intensive care units. Some were from, as I said, various wet lab surfaces. And also, I forgot to mention, there were several office surfaces. And what we saw was that most of the indoor samples had skin microbial communities on them. And in fact, many looked 100% like skin. So here's an example of the incubator surface in a neonatal intensive care unit, which looks almost completely like a skin microbial community. And that makes sense because we know that what's floating around in the air is largely comprised of dead skin from people. And I mean, you know, in the hospital environment. And but then we saw other samples, like here was a button on the incubator for the infant that had some skin community, something that looked like a gut community, and then the rest of it was unknown. So this could be some contamination from changing diapers. Then here are two phone samples that were taken from an office in New York. And these samples have some skin, some unknown, and then they also have this portion of oral communities, which makes sense because it was the mouthpiece on the phone that was sampled. One nice thing about Sourcetracker, or using this Markov chain Monte Carlo approach, is that as you keep reassigning species to different source environments and you keep iterating through this randomized process, you can, once you've done it for a while, you can pause the process and look at the distribution of source environments in your sink sample. And then you can let it go for a while longer, or even restart it completely, pause it again, and look at the distribution. And each time you're getting a different sample from all of the likely distributions. And you can visualize that here. So this is one of the outputs of Sourcetracker. Here's the pie chart, which is showing the average mixture. Here's the distribution of mixtures. And what this tells us, so every column in this stacked bar chart is a different sample, statistical sample that was taken from the Markov chain Monte Carlo simulation. And they're all basically the same. So this tells you that the estimates of the source proportions are very stable here. Then here's a sample that was very low depth, so that it was PCR water, which did have some bugs coming out of it, but they were, fortunately very low, and low abundance. And here, the pie chart doesn't tell the whole story. So the distributions tell you that in most of the samples, the algorithm thought it was just completely unknown. Then in a small fraction of samples, it thought that about half of it might have been gut, or half of it might have been skin. And so here you can see that it's a very unstable distribution. The other nice thing you can do is when you take a snapshot of the contributions of each source environment to the sink sample, you have not only the mixing proportions, but you know how many of the observations of this particular species came from source 1, how many of those observations came from source 2, how many came from unknown. And then you can actually ask the question, what were the bugs that were most contaminating from source 1? Or if you wanted to actually remove all of the contamination from a given source, like let's say you ended up with skin contamination on in some of your samples, if you had a good estimate of the contamination source, you could actually run Sourcetracker and figure out which bugs to pull out, not the entire bug, but just some of its counts to remove the effect of the contamination. So we've run this on a number of data sets now. Here's one example on the ancestral microbiome. So this was some amazing work with some collaborators at the University of Oklahoma, where they had gotten microbial DNA out of fecal samples that were collected in caves. And so these were very ancient human fecal samples that had just been desiccated and preserved for a long time and still had bugs in them. What we didn't know is, did the communities in the fecal samples, these are called coprolites, did they actually resemble a gut community, or had it been invaded by a soil community, or did it have just skin contamination from the people who did the extraction, and so on. So we ran these through Sourcetracker, and we used as the sources some non-westernized human gut samples, compost samples, so it's just a decomposition microbiome, oral samples to look for oral contamination, primate gut, skin in case there's skin contamination, soil in case the soil bugs invaded the coprolite, and then modern USA gut samples, and then of course there's the unknown. And what we found was that most of them, so we had 8 samples, most of them had mostly unknown signatures in them. Not a lot of skin contamination, but 2 of the samples, these were 2 of the best preserved samples, actually had a good fraction that looked like a gut community. And specifically, they looked like a non-westernized human community, or in the case of this Riozape ZA4 sample, it actually was, it looked somewhat like a modern but non-westernized human gut microbiome, and somewhat like a primate gut microbiome, providing a little bit of suggestive evidence that our ancient human microbiome may have been more similar to what modern non-human primates have. Here's another application that I thought was interesting from Vanessa Radara and Jeff Gordon and collaborators. This is a study where they had taken the bugs out of some obese humans and the bugs out of some lean humans, transplanted them into mice, and you've probably heard about this study. The obese donor mice gained weight and the lean donor mice maintained weight. But then what they did was to monitor the bugs in those mice over time. And what you're seeing in this figure is over here, these are the lean mice in this block. And the left half of this block, these are all of the bugs that, that were prevalent in the lean donor. So the lean donor is the source here. In the obese mouse, that's the middle large block. And on the left half of that middle large block, you see the blue bugs. Those are bugs that were estimated to come from the obese donor. So these are assignments, red and blue, made by Sourcetracker for which bug in individual samples came from which source environment. Then they co-housed the mice. So that's the second half of each of those blocks with the yellow or the orange bar at the top. So what happened? Let's see. In the lean donor mice, they kept most of their bugs. There was one or two obese donor bugs that during the co-housing got into the lean donor mice. But the obese donor mice were almost completely invaded by all of the lean donor bugs. So in these samples, Sourcetracker was run on each of these individual samples. So each column is a new sample. And Sourcetracker estimated that all of these bugs had actually come in from the lean donor mouse source environment. And interestingly, when they were co-housed, the lean donor bugs seemed to protect the obese mice from the weight gain. So in summary, microbial source tracking can be done at the community-wide level. You have to use more sophisticated statistical methods because you don't always know all of the source environments well. Sourcetracker can do this. It uses Bayesian methods to deconvolute mixtures of source communities. This lets you identify the contributions of individual species from each source environment. However, it doesn't model any of the temporal dynamics after the mixing. So this would be a perfect model if you took like the PCR products from 3 different communities, 2 known and 1 unknown, and poured them together in some ratio and send them on the sequencer. What it's not a great model for, theoretically speaking, is two environments mixed together, and then they sit there for a week interacting and doing things, and then you do the sequencing. However, we found that in a lot of biological applications, it has still been quite useful, even though there may be some temporal dynamics. And it's available on GitHub, so you can see release 1.0 there, which automatically gives you these outputs, the outputs of the individual contributions of each bug from each source. So that's all for this video.